So, part 3, DNA sequencing. So again, I'm Dr. Dennis Mishler, for anybody just joining us for part 3 here. I'm a professor at UT Austin in Texas in the United States. Okay, I've been working with iGEM for a number of years now. So DNA sequencing is a hugely important thing, and it's something that I think iGEM has not really encouraged enough in the past, and I am very passionate about encouraging it because I think it's a very important best practices. And so when we're talking about DNA sequencing today, we're going to be focused on Sanger sequencing, okay? And we're going to be focused on how to use Sanger sequencing to interpret whether or not your DNA constructs are correctly made, okay? Before I go any further, I want to highlight that there's Sanger sequencing, which traditionally has just been sequencing, but Sanger sequencing, and there's next-gen sequencing. And next-gen sequencing is sort of the high throughput sequencing lots and lots of stuff at once, okay? And there are many different techniques within next-gen sequencing. We're not going to talk about any of that, okay? We're going to focus on Sanger sequencing, the more traditional sequencing, because that is still, I think, most commonly used to look at novel constructs or novel plasmids that you might be making in the lab. This being said, many people, not many, a few people, some people, those with lots of resources will actually use next-gen sequencing on their plasmids. So like they just have lots of money and they have lots of resources and they will do that, okay? That is not the expectation nor the standard practices, I think, right now. That might become standard practices in the future for researchers and scientists, but right now I think most people still mostly just do Sanger sequencing and they don't worry about next-gen sequencing of their plasmids, okay? So Sanger sequencing, we'll talk about what that is in just a second. Let's talk about how we get there. So first off, to sequence your construct, you start by picking an isolated colony from your transformation plate. So I showed these plates earlier. So you can see these circles here. These were colonies that some students picked. You'd pick those colonies, you let them grow overnight. You then take those overnight cultures. Part of that culture, you're going to make a glycerol stock, so you can stock away your sequences. And then the other part of that overnight culture, you're going to actually do like a mini prep, a plasmid prep of some sort to extract out the plasmid DNA, okay? And taking that plasmid DNA is where DNA sequencing really starts from, okay? And as I mentioned in part 1 of the webinar today, sequencing is essentially quality control. You know you have the right thing. So plasmid's purified. What we need to do now is we need to send that plasmid with a primer to get a sequencing result. And the way we do that is we take some plasmid, we take a specific primer, we mix them together, we put them into one reaction tube, and we send that usually in the mail or we'll walk it over to a sequencing facility. And that sequencing facility is going to read what DNA sequences you put in there. Oh, you put this primer in there with this plasmid, and they're then going to go conduct a sequencing reaction for you. Now, primers, this is actually the most important part of doing a sequencing reaction. You have to decide which primer to use. And for a lot of plasmids, there are some standard sequencing primers, but those sequencing primers vary from plasmid to plasmid. And so if you're going to sequence, you just want to figure out, are there any standard sequencing primers for your backbone? You would want to read about that and see what the sequencing facility uses. You could have one plasmid that's really standardized and people use this plasmid a lot, and it could have completely different sequencing primers than another plasmid that people use a lot. So you just have to be aware of that. Commonly though, my students will also just design our own sequencing primers because we might be working with a specific DNA sequence and we're like, yeah, just use your own sequencing primers, that's better. So it just depends, right? But once you have a primer that works, you add it to your DNA template, you send it off to the sequencing core, or sequencing facility, they're gonna go ahead and do the reaction for you. And they're gonna send you back something that we'll call a read. And this read is going to be somewhere between 700 to 1,000 base pairs ideally. And that's going to be DNA sequence. They're going to tell you what that DNA sequence was based upon that primer you gave them. They're going to tell you what's present in your DNA. But each sequencing reaction, each read will only be at most 1,000 base pairs if you're really lucky. And so if you want to sequence a construct that is longer than that length, you're probably going to have to send in multiple different sequencing reactions to look at the entirety of your DNA construct. Each reaction will contain the same template, your plasmid, but will have a different primer, and you'll have to design and plan that out accordingly. So if you're working with a genetic circuit that's, let's say, 1,500 base pairs long, you're going to need at least 2, maybe 3 DNA sequencing primers in order to see the whole sequence to know that it's correctly made. Okay. We're going to really quickly go to, and hopefully I'll share screens correctly. We're going to really quickly watch this 1-minute video on Sanger sequencing. I think it's actually a really nice video. Let me hopefully correctly share everything. And if things do not look right, please let me know. All right, boom. All right, hopefully you guys see this and playing.
The first method of sequencing the genetic code was devised by Fred Sanger. To sequence the DNA, it must first be separated into 2 strands. The strand to be sequenced is copied using chemically altered bases. These altered bases cause the copying process to stop each time one particular letter is incorporated into the growing DNA chain. This process is carried out for all 4 bases, and then the fragments are put together like a jigsaw to reveal the sequence of the original piece of DNA.
Okay, all right. So I love that video because it's so short and to the point. Let me see if we can get this share screen back to the presentation correctly. Boom. All right, and let me see if I got this. All right, did that work, guys? For those, and I can't see anything on my screen. Oh, there he goes. Okay, great. There he goes. Okay. Okay, I'm being told the screen sharing is working again, so great. I'm going to go ahead and keep going, hopefully. Okay, so. Okay, great. So, sorry. Okay, great. So with that, that is how the process works in a very simplistic fashion, right? So you're going to basically take this primer, you add it, you have modified nucleotides, and these nucleotides contain some signal that can be read, and they do chain termination. So they go until they hit the right nucleotide, stops, and that gives you a signal that tells you which base was present. And so you can do a fraction of ratio of mixture of chain terminating nucleotides and chain continuing nucleotides so that you can see the entire sequence. And what you're going to get back from this process is going to be something we refer to as a chromatogram. Essentially, it's going to be a record of the signal that was detected during the sequencing reaction, as well as their algorithm's prediction for which nucleotides were actually present. And so here's an example chromatogram. You can see the nucleotides down here. This is what the program predicts is present. And it's using these peaks, these 4 different colored peaks. It's using these peaks to tell you what nucleotide is present. Each color represents or is corresponding to a specific nucleotide. For example, A's are green, where we got G is black, C is blue here, and T is red. And that's pretty much universal. I think that's always going to be the color scheme that you get back. And so you can look at this chromatogram and basically just figure out what your sequence is. And you can either do this yourself or you can rely upon the algorithm to tell you what it is. So what I'm going to do here is I'm going to go ahead and transition over to benchling. We're going to change screen sharing again and give you guys a little run through of working with a chromatogram. And we'll use this to look at a sequence, and I'll talk about good and bad things on that sequence, okay? And importantly, during this time, we're going to talk about how to interpret signal, noise, and whether or not you have an actual mutation in the sequence you're working with, okay? So let's go ahead and new share. I think this will be right. And boom. Great, okay. And you guys see benchling now, I'm hoping. So, okay, great, perfect. So I'm gonna go ahead and remove my camera so I can sit down here and talk to you guys. Okay, so what we're seeing here is a benchling sequence. And this is actually a sequencing result. So from the sequencing facility we use, they sent us this back. And when they sent this back to us, you can see the DNA sequence is just here. There's no chromatogram though, right? I'm just looking at the sequence as shown on benchling. So let's go ahead and open up the chromatogram. So I'm going to go over here to this alignments tab, and the sequencing result comes with its own alignment that includes the chromatogram. Oops, let me go ahead and delete this off here. I was working on this earlier. Okay. Uh-oh. Okay, that should, uh-oh. Let's see, let's close that out. Uh-oh, sorry guys. Let me refresh this. Okay, so I clicked the button and it froze. So what we will see here in just a second, I'm gonna go back over here, great, is I'm gonna delete this real quick, get rid of that one. Okay, great. So what we're gonna see here is just the chromatogram and the DNA sequence predicted. So the top is the DNA sequence predicted, and this is the chromatogram down here. And you can see as we go along, the peaks get smaller, and that's because of the chain terminators. As you go further into the reaction, less DNA sequence is full length or going further, right? Every time you terminate, you lose one product, right? And so at some point, it gets so low, you can't really see it. And so we can use this bar here to just increase the signal there. If we want, you can see what the signal looks like. And eventually the signal gets so low that it just sort of stops, all right? And you can see down here, there's mostly just ends. Ends mean any nucleotide, or you can't really make a prediction. So whenever you see an N on a sequencing result, it means the data there is not very good, all right? The algorithm had a problem determining what nucleotide or what base was present. And so it just goes, I don't know, N, could be anything. But here you can see we're about 750, 800 nucleotides into the read, and the quality is degrading pretty quickly here. So we can't really go any further. We can't use any of this information here. So we have a good readout though to about 750, so that's great. And we can go back to the beginning, and you can see how high the peaks are here. So we're gonna just change this amplitude modulator, bring it back down, boom. And here you can see the first 10 or 20 nucleotides also ends. And so when you're doing your sequencing reaction, what's really common is the very beginning, the first 20, 30 nucleotides, the system is just trying to get everything under control. And so you often can't look at that data either. You really want to start a little bit later. And so here's where we're starting. And you can see the reads are really, really crisp, really clean. You have these nice peaks. They're very standard, very by themselves. There are in a few places peaks that are sort of clustered. So right here, you can see these two are really close together, overlapping a little bit. Okay. So this though is a good read out to about 700 base pairs. So it's something we can work with. That's important. And what I want to do now is I actually want to align my reference sequence to the sequencing result. So I'm going to go in here, I'm going to add, I'm going to search for my reference file, which I know is this. It's something I use a lot. So I'm going to use this reference sequence here that I have with my students. We're going to hit realign, and we're going to see the reference sequence will pop in here. And the reference sequence is this middle one that has all this red. Okay. I'm going to highlight the fact that the reference sequence, if I click over here, the reference sequence is 7690 bases long. That's really long. My sequencing result was maybe, what, 750, 800? And so all this red you see down here is simply saying this information does not match the blank information up here. So, right, there's nothing up here on the top. So that's why it's red. Just there's nothing there. We can ignore all that. But let's go to where the sequencing result begins. The sequencing result starts right here. And so we can compare now the reference file, which is this middle one here, to the chromatogram to see if we think the chromatogram is correct, if the sequencing result looks good. Real quick, the reference file has this these amino acids here. This is just the reading frame translated. It has an annotation here of the gene. It starts there. And you can see the gene starts way over here at about 3,200 or so. And we're looking at it way over here, okay? So let's go ahead and look at the sequencing result. First, red means it doesn't match, okay? So red, it's not matching because these are ends, so there's not enough information there. We then start to look here, and you can see once we get to here, we start to see very few reds. So the reference file is matched by the sequencing result. And everywhere we see that there's not red, it's a good match. That's perfect, great. That good matching is represented by gray down here. So if you look in here in this bottom part of the window, you can see all the gray. That means the reference file and the sequencing result match each other. That's a good sign. Let's go back to the beginning though, because we had a couple of reds. We have a red here and we have a red here. And so this is where understanding the chromatogram is really important. Are these mutations, something's wrong, or is this just an artifact of the sequencing? Was there an error or something, or the sequencing result was a little unclear? And so we had to figure that out. So this one's easier to deal with. We're gonna look at this one here first. It's an N, which means the algorithm was unsure. It was not sure what was happening. The reference file though says it should be an A. So if we look at the chromatogram peak down here, we can see T's are red, blue, there's a C here, then there's an N, and then there's a black peak, which is G. Well, there's another peak in here right here. If I click over here, you can see it more clearly. There's this blue C, and then there's actually a green peak here. They're just aligned kind of closely. And green is A. So you can see all these A peaks over here, right? So there's actually at this N position, there is an A peak. It's a green peak, it's there. It's just that the algorithm wasn't able to deduce that. So I would say this N is actually the A. That looks fine to me. I'm not worried about that at all. That looks good. But this one right here is a little bit more worrisome. Here you can see the reference says there should be 2 A's. We should see 2 green peaks, but we only really see 1 clear one down here. And so it's not clear to me if this is a mutation or not. It's possible we're at the beginning of the sequencing read, and so the data is still a little wonky. It hasn't yet figured out what's right. And so we need to probably resequence this, right? If I wanted to work with this sequence confidently, we would probably need to resubmit it to figure out if this is actually a mutation or just an artifact of the sequencing result, okay? The other thing I want to point out is there's this one red bar right here, and we have to take a look at that. We have to know if this is a mutation or not, because it's in the middle of our gene. And you can see again, it's an N, which means the algorithm was unsure. It should be a C. And so we have to increase the amplitude here because the signal is very low, right? We're further into the reaction, the signal's lower. And as we do this, you can actually see there's a blue peak here. It's actually a C, right? Blue is C. It's just this peak is a little bit lower than the peak surrounding it. And thus the algorithm was unsure about what to do with that peak. But that's a C. I'm confident that's correct. All right, we look at it. There's nothing else really around there. It's probably correct. So I think that this is just a C that's hard to read by the algorithm. And if we go a little bit further, we'll start to see we start to get a lot of red in positions, right? All these positions up here are red. And this is because the signal is so low that we're now at this signal to noise ratio where the algorithm is no longer confident that signal is signal. The signal may just be noise. And so we can't really interpret anything from this part of the read. So our read probably really ends about here. Once we get to here, I wouldn't interpret anything. So I would basically cut off the read about here. I think anything to the left here is good. Anything to the right, I would ignore. We need to have another sequencing result to compare and look at that part of the sequence. So we have to design a new primer. Probably I'd design the primer somewhere around here. This primer would be the new sequencing primer, and then the new sequencing read and a new reaction would start somewhere around here, allowing us to look at the rest of the sequence, okay? So that's a little bit about how you take a sequencing result, do an alignment, and then interpret the data, right? You need to look at the signal, the noise, and see what's happening, okay? So let me go ahead and escape out of this, and let's go back to the webinar if I get everything done correctly. I may have to change screens in a second, so just a heads up on that. Okay. And then if you guys can let me know what you see or don't see. Okay. Great. Okay. Do you guys see the presentation correctly? I'm going to keep moving forward, assuming you do. Okay. Oh, you see multiple windows again. Okay, 1 second. There we go. All right, that should be good. Okay, so we went through this. We went through the benchling procedure. So sequencing summary and questions. So let me summarize some things real quick for you here. So hopefully you guys learned about DNA sequencing and you saw a little bit about how to interpret your results. When working with the DNA sequence, move that back up there, it's absolutely vital to make sure your sequence is correct, right? Even if you see the right phenotype but you have a different sequence, that could tell you that something is off or give you different results, right? You never know when a mutation might change the rate of transcription, the rate of translation, or change the functionality of the protein. So you need to be very careful about mutations, okay? Mutations happen all of the time in genetic circuits. So even if you got a sequence from somebody else in another lab, that doesn't mean it's correct. It could be mutated because of the time it took to grow. So you want to just resequence it when you get it, all right? There are lots of resources online. I did show you that one video that was really brief. That link is here again. There's a second video in the links if you want a little more involved explanation for Sanger sequencing. If you have additional questions, I want to take some right now, but we also have our office hours today. So if you came through these webinars here, the 3 parts today, and you're a little confused on something, pop in. The office hours at 1 p.m. Boston time. I'll be there for at least the first 30 minutes. Come on by, take a look. If you're not sure, you need to contact me. Otherwise, send us an email at this address here. Just ask for me. Again, my name is Dr. Mishler. Happy to answer your questions there. So with that, let's look at the questions here and see what we have. All right, great. So I have a bunch of questions already. So I'll read off the questions and comments and then give some answers. And if you have more, just keep them coming. So first one, somebody said, Alexander said he liked the jigsaw example. I do too. I think that's a great 1-minute video. I think it's really clear. If you're new to it, it might have been a little confusing, but if you go back to it once you've seen a little bit more and look at it, I think it makes it really clear what's happening. You're getting these 4 different sequencing reads of where the different letters are, and then you're able to combine everything together. I think it's a really nice, quick way to describe it. Aparna, you've been great. You've been asking questions, giving comments. I love that. Can we have a separate class for chromatogram where things will be explained from scratch? I think if you can make the office hours, that seems like a great use of office hours time. Okay, I would be happy to go over that with you. It is about 11 a.m. right now, Boston time. So in about 2 hours from now is when we'd have the office hours. If you can let me know if you can make it or not, we can do something there. If you can't, let me know and we can figure out a way to go over chromatograms separately, okay? Yeah, so chromatograms are really important. So if you guys are having issues with that, just let us know and let's see if we can do office hours or something else. Hassan asks, what are the other ways? Oops, something happened here. Sorry, something just popped in. What are the other ways to check if there's really a mutation? You really have to use the sequencing result. And what you should see is a clear peak that says C, and then your reference file says A. So if you have a very clear peak that indicates this is definitely not the nucleotide the reference file has, mutation. If you see things are missing, so in the example there I had a potential deletion, right, and A was missing, that would be another example. Those are kind of the 2 main ways to do it. There are some other ways, like if you just see that you don't get the right sequence, right? If it should be green and it's not green, there's something wrong. But in terms of the sequencing results, that's generally what we're looking at. Okay. Let's see here. If we perform Sanger sequencing multiple times, will that help? Yes. So Michael gave an answer, but yeah, generally, if you have, if you repeat the sequencing result, sequencing reaction, and you get a different result, it tells you there's an error in one of the 2 sequencing reactions. You can alter where the primer is located to help you a little bit with the resolution. But sometimes if it's like in the middle of the sequence, sometimes you just resend the exact same sequencing reaction in a new tube, and then that'll clear it up, right? Maybe there's just a little error in the machine that one time, or the reaction was just a little off. So sometimes you can just send the exact same primer template mix and get the clean result, but it just depends. Sequencing reactions are also done in large scale, in bulk. And so a sequencing facility, it may have just had a bad run. That happens sometimes. And so every reaction that was run at the same time might be 20% less clear than normal. And if that happens, you were just unlucky. And so you may have to ask them to rerun the sample for you. Next, we have a question about preparing a sample to be sent for Sanger sequencing. The best answer to how to prepare a sample for Sanger sequencing is to find the sequencing facility you're going to work with and see what their procedure is. We have a very clear procedure in my group that we use, and it's very simple. It's you add this much template at a certain concentration, you add this much primer at a certain concentration, you add water, send the sample in. Those concentrations, though, might vary from facility to facility. And so if you're working with company X, you need to see what their procedure is and follow theirs. And it should be online. It should be very clear. And it's going to be like, send us this many nanograms of DNA, or send us this many nanograms per microliter of DNA. That's going to be the main part. And then they'll tell you the primer separately. Okay. Please upload slides soon. We will, Alexander, as soon as we can, I promise you, we'll put them up there. All right. Aparna, you think you can make it at 1 p.m.? So great. We'll just ask for me there. Happy to answer then. Great. We have a sorry if you answered this already, but to sequence the next piece, do you use just a different primer that matches the end of the piece you have? Yeah, so I kind of went over this just briefly on benchling, but let's say my reaction goes to position 750. My sequencing read out to 750 looks really good, but now I want to look at 750 to 1500. Well, I need to design a new primer, and that new primer probably needs to be around position 700. And the reason for that is that first 20 or 30 nucleotides will give you really hard to interpret data. And so then the second sequencing result will start really around 730, 740, 750, giving you some overlap from the first sequencing read. And then it'll keep going from 750 to hopefully 1200, 1300, 1400, and so on. So yes, exactly like you said, design a different primer that matches not the very end, but maybe about 50 nucleotides before the end of your first read. That would be how you would design that, okay? So hopefully that answers the anonymous person. Okay. All right, cool. Aparna, again, just show up on the thing. You sent me your email here, but if you want to contact me, please email me on the address here on the on this final questions and answers sheet. I will not be able to record this email here, so please send that to me. Okay. Great, Satino, I'm glad you've liked the webinar. That's always great to hear. I love getting feedback from you guys. If there are more questions, please let me know, folks. I see there may be one more here. Hassan, you've asked, do you always measure the concentration of DNA using a nanodrop? Ah, this is a real, okay. So this gets down to how do you quantitate the concentration? How do you measure the concentration of your DNA? Many people use nanodrop. I am not a huge fan of nanodrop. Nanodrop concentrations are regularly off by a factor of 2. But nanodrops are widespread and widely used. So if you have a nanodrop, it is better than nothing. Do it. Use the nanodrop, send those concentrations in. Nanodrop, though, is regularly off by quite a bit on the concentration of DNA. We actually use something called a qubit, which is a fluorescence-based way of measuring or quantitating how much DNA you have. That gives way better results in my experience. And so we will get our DNA concentration using the qubit and then send it in using that. So here would be an example. We need a final DNA concentration of 20 nanograms per microliter in a total of 10 microliters. Let's say that's just what we have to send into the sequencing facility, okay? We quantitated our DNA, and our DNA is at 200 nanograms per microliter. So now we have to dilute the DNA to 20 nanograms per microliter in the volume they've specified, 10 microliters, okay? So we have to do a dilution, and we do some math to figure that out. So yes, we always send in the same volume of DNA, but sometimes we have to dilute our plasmid into water before we send that in, okay? So it's really important to look at the sequencing facility's specifications for what they need from you before you send in your samples. And again, if you have nanodrop, use it. It's better than nothing, but nanodrop is frequently off. And so I've seen sometimes the students will send in nanodrop samples and they'll tell us the concentration is just way too off. And so I always tell my students, if you have the time, use qubit instead and use that concentration. That will generally be better. So it just varies. Okay. All right. So 1 second, I got a couple questions here. So Alexander asks, the quicker you send a sample in for sequencing, the less chance for mutation. Not really true. It's more about your cells growing. If you let your cells grow for a long time, if your cells sit in the fridge or the freezer for a long time, those will be more prone to mutation. Once the DNA has been isolated, if you freeze that DNA down and you keep it frozen, I would not expect a lot of mutations. So it's really about the cells and the cells growing. That is where you're going to get a lot of the mutations creeping up. So if you grow an overnight culture and you put it in the fridge for a week and then you take it out of the fridge, that will likely have more mutations than if you had isolated the DNA right after you grew up the overnight culture. So if you grew an overnight culture, you come in the next morning, isolate the DNA that day if you can, freeze it down, and it should be good for a very long time. But if you take the cells and you put them in a fridge for a while and then later you isolate the DNA, you're far more likely to get a mutation. Okay, I think that's the right way of looking at it. Great, good. You know, I'm going to skip a couple of these questions here just because of time here. Nanotech, sure, you can use nanotech with DNA sequencing, totally agree. For transformations, are you sometimes using transfection via liposome? We don't. We only work with bacteria. But yes, people with mammalian cells, absolutely. With mammalian cells, yes, you got to do stuff like that. What works with bacteria is often very different from than what works for eukaryotic organisms, yeast, C. elegans, mammalian cells, all that type of stuff. And you have to use very different procedures based upon the organisms you're working with. Okay, so there's bacteria and there's eukaryotes. And within bacteria, there's kind of E. coli and then everything else. And so you really just have to be aware of the organism you're using and the differences in the techniques for each organism. Okay. Quick, rapid refreeze. So, you know, I don't like freezing and thawing things. That's just always an issue. If you have to do it, it's not a big deal though, right? Like you can freeze and thaw something once, but if you have to do it repeatedly, you're more likely to cause mutations. That's true. So try not to, but it's not the end of the world. That's one reason why what I usually do, if I have a plasmid that I want to keep, I'll keep it frozen at -20. And if I want to use it for PCR and such, I'll make a separate tube that's my dilution tube or my working solution. And that one will get frozen and thawed repeatedly, and then I'll toss it. And then I can always go back to my frozen stock, which is kept always cold, and take a little bit out of that to make a new working tube that I work with. Okay. All right. All right. Hopefully I've answered everybody's question. Thank you guys so much. You guys were a great audience. I love the questions. If I didn't answer your questions, shoot me an email. Email address is right down here. Shoot me an email, or come to our webinar, or not webinar, our office hours. Office hours will be in about an hour and a half, and we'll be happy to answer your questions there. I'll be there for at least the first 30 minutes before I have some meetings with students. Okay. All right. Thank you, everybody. Have a great day. Please stay safe. I hope COVID's not ravaging your country like it's ravaging mine. So please stay safe, guys. Best of luck to everybody. Talk to you guys later. Okay. Bye.